Hello and welcome to your Chapter 8 lecture video on the muscular system. So similar to the skeletal system from last week, there is a separate PowerPoint in the lab folder that has slides which go over the individual muscles. So make sure you're also looking at that. Also in the lab folder there is a specific list for the muscles that you'll need to know for lab. We'll discuss that when we're in person, but I wanted to let you know that there are other slides associated with this lecture. I'm not going to record anything about them, but they have all the muscles you need, all of the muscles that we kind of cover in this body, plus a couple more with their origins, insertions, actions, all that information. So let's just jump into this. This lecture is more on some of the basic anatomy and physiology of your muscles. So basic introduction to your muscular system. Similar to bones, muscles are organs. They have multiple tissue types: nervous tissue, blood vessels, muscle tissue, obviously, and connective tissue that all help to produce their functions. Some muscle actions, things like walking, breathing, pumping blood, moving food through the digestive tract, these are all functions of your muscles. You might mostly think of them as, your skeletal muscle that's doing the actions of moving your joints, but as we can see, there's a lot of other functions in muscles that we're going to get into. Three basic types of muscle tissue: skeletal, smooth, and cardiac. We're going to mostly focus on skeletal muscle in this lecture. We do discuss a little bit of smooth and cardiac muscle later towards the end of it. So there are over 600 skeletal muscles in the body. We're first just starting kind of with the basic structure of these muscles. They're all pretty similar. They have these connective tissue coverings around them. And when you think about your muscle, a lot of times we'll talk about muscle fibers. So if you hear me say that, that just means a muscle cell. A muscle fiber is a muscle cell. And if you kind of sit here and just kind of use one hand and just kind of start to massage the muscles in your arm, you might feel it starts off as one kind of big lump, but then as you rub into it a little bit, you might start to notice there's some bands that you can palpate. Those aren't the fibers because muscle cells are microscopic, but you can start to kind of feel the texture of your muscles, and you can see it has this kind of banded appearance. And so what's covering all of that is this fascia, this connective tissue that's surrounding your muscle and then also dividing it into these smaller functional groups. That connective tissue goes beyond the muscle. It starts to create the tendons that then become the periosteum of bones. So if you remember, tendons are structures that connect muscle to bone, T-bone, like a T-bone steak. Your muscles and your bones are connected by tendons. The connective tissue covering around your muscle becomes tendons. Tendons become the covering around bones. So that's how integrated these structures are. There's also other tendons. So usually when you think of a tendon, you're thinking of very small, specific ones attaching, a muscle to a bone. There are these things called aponeuroses. We have several aponeuroses throughout our body, and these are broad tendons. Rather than being a specific, smaller, isolated tendon attaching one muscle to a bone, these are going to be attachment sites for multiple muscles, typically across a broad range of area. So, for example, the top of your head, across your low back, in your calf and your forearms, there are these broad sheets of connective tissue. So let's look specifically at the coverings around these muscles. So what we first have is the epimysium. That's the outer layer of connective tissue around every muscle. So epi, upon, right? Mys, M-Y-S, this is the muscles. So this is the covering around the muscle. And it then starts to subdivide into smaller compartments. That's called the perimysium. So peri meaning around. So within your muscle, you're kind of in these groups called fascicles. Fascicles are bundles of muscle fibers, and they're wrapped around with perimysium. And then around each muscle fiber, around each muscle cell, is a connective tissue layer called endomysium. And again, your epimysium, perimysium, endomysium, they are all related to each other. They're continuous structures from each other. We're just getting smaller and smaller. So the smallest organizational unit is the muscle cell, the muscle fiber. Multiple fibers come together to create fascicles. And then multiple fascicles are bound with the perimysium to actually create the entire muscle, and that's your epimysium. So here's a picture of that. I would start up here with the bone, where it connects with the tendon to your muscle. You can start to see this epimysium. First, there's just that white fascial layer, and then you can see this kind of thickened, let me zoom in a little bit. You can see it has this little thickened layer here of the epimysium wrapping around the whole muscle. And then each one of these bundles is a fascicle. And every fascicle is wrapped with perimysium. And then when you pull your fascicle out, so here's your fascicle, every fascicle is a bundle of muscle fibers. And every muscle fiber is wrapped with this endomysium. That's that layer that's directly around a muscle cell. Now, what you're seeing here again is a muscle cell. Each one of these is a muscle cell. But you're like, I'm sure you're like, well, what are all these things that are filling that are stuffed in there? When we talk about a cell, I know normally when we study them, it's a nice round cell, and you see the Golgi bodies and the vacuoles and the lysosomes and the peroxisomes and all of those things. That is a general cell. We are looking at a specific specialized cell here. So each one of these kind of tubular shaped bundles, and this is why we call them fibers, because they're in this kind of fibrous shape. If you work with yarn at all, if you ever worked with, or if you haven't, go find some yarn. It looks just like a strand of yarn, one solid strand. But if you start to unwind it and peel it apart, you can peel apart the individual fibers within it. And so that's what we're looking at here. Is that each one of these fibers is filled with all these little strandular structures. So rather than having all that empty space, all that cytoplasm filled with, like I said, endoplasmic reticulum and Golgi bodies, I'm running out of words to say, but instead of being filled with those, it's filled with these filaments, with these myofibrils. Okay, so each one of these myofibrils is what's filling up the space within a muscle cell. The myofibrils themselves are made up of myofilaments. So when we talk about muscle contractions, it's actually happening at this level, at this filament myofibril level. The myofibrils that pack, that fill up the space within a muscle cell, they themselves are surrounded by what are called sarcoplasmic reticulum. So instead of endoplasmic reticulum, we have sarcoplasmic reticulum. And go ahead and get used to this prefix of sarco. Anytime you see sarco, it's referring to your muscles. We'll talk about sarcoplasm, sarcolemma, all of that. So sarco refers to your muscles. So the sarcoplasmic reticulum are surrounding your myofibrils. Then there's some other tissue structures that kind of cover that. And then again, it's all wrapped in epimysium, and that's what creates your muscle fiber. Okay, we're going to get into those structures a little bit more, but if you ever get confused, I would definitely save this slide, save this picture. It's one we're going to use in lab. It's one I'm probably going to quiz you on, because understanding the structure of how, kind of like this nesting doll structure of muscles is going to be crucial. It's also going to come back when we look at the nervous system. Okay, so looking at muscle fibers specifically, so each fiber is a long cell that is a long cylindrical muscle cell. This is what stimulates the force to pull on joints, to pull on attachments, and cause movement. The cell membrane, rather than having, we talk about plasma membranes, cell membranes, whatever, the covering around a cell, we call it the sarcolemma. So the sarcolemma is the plasma membrane. The cytoplasm, rather than just cytoplasm, we talk about sarcoplasm. Are they the same thing? Yes, but you wouldn't call a cell in your, a normal liver cell, you wouldn't talk about its sarcoplasm, you would talk about its cytoplasm. It's just to be specific. Honestly, it's probably some anatomist that studied muscles and wanted to make sure that they had their own unique name, and so that's why you have to learn it as sarcoplasm and not just cytoplasm. Cytoplasm, I did it too. The sarcoplasm of a muscle cell is going to have a lot of mitochondria and nuclei. There's going to need to be a lot of production of ATP, and so it makes sense. As we said, the sarcoplasm is also packed full of these myofibrils. And myofibrils are bundles. They are bundles of your thick and thin filaments. So you saw on that slide, it talked about the myofibril being filled with these filaments here. That's what we're looking at. The thick filaments are mostly proteins called myosin, and the thin filaments are also a protein called actin, a thinner protein, obviously. But it's wrapped, that actin protein is wrapped with troponin and tropomyosin. And we'll see that these two act as regulatory proteins. They wrap around the actin and have to be kind of moved out of the way before muscle contraction can happen. It's one of the reasons why you're not just always in a state of contraction, is because these proteins are in the way and blocking the binding. It's the way that these thick and thin filaments are laid around each other, that organization of them, that give rise to the striations that are characteristic for skeletal muscle. Okay, looking more at the myofibrils and even more specifically the sarcomeres. So a sarcomere is the contractile unit of a muscle. You will hear that over, it's a very kind of common phrase to use. A sarcomere is the contractile unit of a muscle. So a muscle doesn't contract. It's not like the muscle fiber is one long contractile unit and the whole muscle fiber contracts at once. Little sections of each muscle fiber, of each muscle cell, that's what's going to be contracting. It's these sarcomeres contract bit by bit, and that's what actually generates greater force. The sarcomeres have some boundaries to them, some structures that we're going to look at here, and then we'll see a picture of it in the next slide or two. So a sarcomere goes from Z line to Z line, right? Z is the end of the alphabet. It's the boundaries of your sarcomere, from one Z line to the next Z line. There are those striations that are, again, part of a feature of muscles, of skeletal muscle, and those we actually talk about the I bands and the A bands, the light and the dark bands. The light bands are just your thin filaments, just your actin, and those are attached to the Z lines, A and Z, right? A and Z attached to each other, end of the alphabet, beginning of the alphabet. And then the A bands, so don't get the A bands confused with your actin, the A bands are in the middle. These are dark bands that have overlapping thick and thin filaments. I'm sorry, it's I on the outside, then A kind of, I say it's the middle because then the actual middle is the H. But A bands are in between the I band and the H zone. Center of the A band is the H zone, which is just myosin. And then the very center of your Z line, of your Z discs, you'll hear that, is your M line, M for middle. And those are just myosin. So M, middle, myosin. Let's look at a picture. Much easier to understand. So here you go. You have, this is an entire skeletal muscle fiber over here. So it's the entire skeletal muscle fiber. You can see the mitochondria, and then each one of these myofibrils. And if we pull a myofibril out, you can start to see it has these bands of thin actin and then thicker myosin. And each one of these kind of squiggly lines here, those are your Z lines, those are your Z discs. I say both. I usually prefer Z discs, but I'll try to remember to say Z line. Each one of those is delineating your sarcomere. So there's a sarcomere, there's a sarcomere. You can see the beginning of a third one right there. The ends are your Z lines. The parts that just have your thin filaments are your I bands. Where the thin and the thick overlap, it's the A. And where you just have the thick, it's the H. And then in the middle of your sarcomere is the M line. Okay? And so what happens is that when the muscle contracts, your Z lines are getting pulled together. So they actually shorten, the sarcomere shortens because these thin filaments that are anchored to your Z line are being pulled in towards the M lines. Here's just another depiction of that. You can see this M line here with the thick myosin filaments, those thinner actin filaments, right along there, those kind of thin actin filaments there, and then the Z line that they're anchored to. And the Z line is what moves. The M line does not move. So now let's look at the sarcoplasm within a skeletal muscle fiber. So underneath the covering, underneath the sarcolemma, you have this sarcoplasmic reticulum, these channels in tube system. Remember, reticulum kind of refers to this tube network, if you want to. And the sarcoplasmic reticulum, they're filled with calcium. We'll see that. They're filled with calcium. I'm sorry, yeah. They're transporting calcium into the T tubules. So as you go into as you go along the sarcolemma, you have these invaginations, these dips down into it. Because the sarcolemma is the covering, but we want to go inside the muscle fiber. And so these T tubules, that's what they call out invaginations, these holes, these tubes that go down into the actual muscle cell itself. The T tubule lies between two cisternae. That just refers to like a holding chamber. A cistern, right? You might have heard of a cistern before that just like holds water or something. That's what this is. Each T tubule is within these two cistern of the sarcoplasmic reticulum. T tubules open to the outside of the muscle fiber, whereas a sarcoplasmic reticulum are more internal. We also hear that's called a triad, where you have the T tubule and the two sarcoplasmic reticulum. That's the triad. The SR and the transverse tubules activate the muscle contraction when the muscle fiber is stimulated. So essentially what happens is you get the neurological signal to contract your muscle. You send you think about bending your elbow and it happens. What's happening is that neurological signal, that electrical signal is traveling along the sarcolemma, along the outside of the muscle. It then goes down into these T tubules. And then that electrical signal then stimulates the SR, the sarcoplasmic reticulum. And so by traveling through the T tubules, activating the sarcoplasmic reticulum, it releases calcium, and that's what causes a muscle contraction. We'll get into those details. But here you can see this a little bit more. You have the sarcoplasmic the sarcolemma is this kind of outer covering around the muscle cell. And you see these holes right here. These are all your T tubules. So an invagination is a hole, tube, essentially, into the muscle fiber. So without that, the signal wouldn't actually be able to go into the muscle cell. The electrical signal would just stay on the outside of the sarcolemma. So this is how that electrical signal can travel into the cell. And as it travels into the cell, so these are T-tubules right here, all these little kind of projections up. As it travels down into the cell, it then encounters the sarcoplasmic reticulum. And so the sarcoplasmic reticulum are covering your myofibrils. And once they get stimulated with that electrical signal, it will activate them to release their calcium that then allows a muscle to be contracted. So sarcolemma, T-tubules go into the muscle fiber, activate the sarcoplasmic reticulum. So let's talk about the neuromuscular junction, because for a muscle to be stimulated, it happens at the neuromuscular junction. So neuromuscular, where your nervous system and your muscular system come together. We also just abbreviate this NMJ. So a skeletal muscle fiber will only contract once it's stimulated by a motor neuron. Sometimes that happens voluntarily or involuntarily, right? Skeletal muscle is voluntary, but every now and then you just get like a random twitch or something. That twitch still comes from a motor neuron. The skeletal muscle fiber is functionally but not physically connected. And that's called a synapse. So it's not like the motor neuron is stuck right up against the muscle cell. There's a little bit of space. There's a small microscopic gap between those two, but they do still communicate with each other. And they use neurotransmitters to communicate. So something important to understand about the nervous system is that the nervous system uses electrical signals, but the body can't, when you go from one structure to the next, there has to be a chemical mediator, a chemical translator. So in this case, we see this electrical signal coming down a motor neuron. It then turns into a chemical signal where at the neuromuscular junction, where those motor neuron and muscle fiber meet. Instead of being an electrical signal, it then stimulates the release of, we'll see, it's called acetylcholine of neurotransmitter. That neurotransmitter then binds to the motor neuron, it binds to the muscle fiber and is turned back into an electrical signal. So it's electrical, chemical, electrical. And you need that chemical mediator for that signal from the motor neuron to talk to the muscle fiber. So at the end, the distal end of the motor neuron, it has a lot of mitochondria because it's creating, it needs that to generate the energy for releasing its synaptic vesicles and bringing them back in. Synaptic vesicles, so this is just describing that these storage pods are at the synapse. And they contain that acetylcholine, that neurotransmitter. The spot where the motor neuron and the muscle fiber meet is called the motor end plate. This is just a folded region of the sarcolemma. It's kind of like a bumpy region of the sarcolemma, and that just allows there to be more surface area at this space, so that way you can make sure you have optimal stimulation. If it was just flat, it wouldn't be as many opportunities for the neurotransmitter to bind. Motor end plate is going to have those specific receptors for the neurotransmitter as well. Once an electrical impulse reaches the end of the axon of the motor neuron, so if you remember from that tissues lecture, we looked at nervous tissue. The axon is that long structure that's leaving the neuron body and taking a signal away from it, axon away. So once that electrical signal reaches the end of the axon, that electrical signal is actually going to change the membrane potential, the membrane charge of the motor neuron, and that then causes this release of the synaptic vesicles, and they get released into what's called the synaptic cleft. That's just the space between the neuron and the muscle fiber. So the synaptic cleft, they also hear called the synaptic gap, truly. That's just the space between, like we said, the muscle fiber and motor neuron aren't stuck together. There's a gap. So that's what this is. And that's where the neurotransmitter gets released. That neurotransmitter then starts to diffuse and kind of float across that cleft, that gap, binds to the motor end plate, and then that initiates another electrical signal. So electrical from the motor neuron, chemical across the gap, and then electrical again as it goes down the sarcolemma. Here's a picture of that. So you can see this is your motor neuron, mitochondria, and synaptic vesicles, and they're filled with that neurotransmitter. Once the electrical signal comes down the motor neuron, these synaptic vesicles start to bind with the membrane of the motor neuron. They're released. they release their neurotransmitter, and then you can see here's this motor end plate, this kind of very folded, tightly folded sarcolemma, portion of the sarcolemma. And along here, it's we can't really see it in this image, but there's receptors all lining this sarcolemma, waiting for the neurotransmitter to bind. Once the neurotransmitter binds and it activates the sarcolemma, that signal gets sent along the sarcolemma. It goes down the T tubules and activates the sarcoplasmic reticulum. So hopefully you're starting to see how this kind of all stacks on top of each other. So looking at a skeletal muscle contraction, I'm kind of putting it all together now. So there are several events that you have to have for a muscle contraction. They all result in the shortening of the sarcomeres. We said that's the contractile unit. And as the sarcomeres shorten, they start to pull the muscle against its attachments. That pulling force is because you have the binding of the myosin and actin molecules, your thick and thin filaments, latch onto each other and they shorten. As they shorten, it's an increasing the overlap between actin and myosin filaments as they slide past each other. Shortening the muscle fibers results in shortening the entire muscle, which pulls on the attached body part to cause movement. So each of these little sarcomeres is going to shorten on its own, and then collectively you end up shortening and engaging your whole muscle. So looking specifically at the role of myosin and actin in this, myosin is two strands twisted together, and they have these heads that project out. So the myosin heads are very important. And together they form the thick filament. Actin has to have troponin and tropomyosin associated with it to create the thin filament. So actin is what actually has the myosin binding sites. The myosin heads are going to attach the binding sites on actin once the troponin and tropomyosin have been moved out of the way. So here you can see this thick myosin filament with its projecting heads. You can see the actin wrapped around troponin and tropomyosin. It's got these regulatory proteins that are hiding these. You can kind of see those little spots right there. Those are the myosin binding sites where the heads want to attach to. So we have to get those troponin and tropomyosin out of the way to expose the binding sites for the heads to attach. Once the heads attach, you see how they're kind of angled outward. Once they attach, they actually change their shape and they get drawn inward. So they kind of cock in, and that pulls the thin filament, which then pulls on the Z-disc, which then shortens the sarcomeres. So we call this the sliding filament model of muscle contraction because we're talking about our thick and thin filaments sliding over each other, and that's what causes the muscle to shorten. We call where the myosin head attaches to the binding site a cross bridge. So the cross bridge formation is a crucial part of a muscle contraction. Once the head binds, it causes it to bend, pulling on the actin, moving it towards the center. Once it moves it to the center, it then can release it, attach to the next binding site on actin, and pull it even shorter. At a certain point, there is a maximum overlap. There's a maximum shortness of contraction in the muscle. If you need to be stronger, then we'll see what's called recruitment, where your body starts to recruit more fibers. But you can use, if you don't have any weight in your hand and you just bend your elbow, you can shorten those fibers to their maximum degree. If you were to put something in your hand and then shorten it, you would be engaging more muscle fibers to shorten the elbow. But the body will always use the least amount of force necessary. So if you don't have anything in your hand, and I'm just going to throw a number out there, because you have millions of muscle fibers, and depending on the muscle, you can have hundreds of thousands of muscle fibers. We're going to use simple. Let's just say there's 100 muscle fibers in your bicep. They're not, they're more. But if there's nothing in your hand, you might only use 10 muscle fibers to bend your elbow. They're still going to be shortening to their deepest extent. Overlapping those filaments. But if you were to, let's say, put a 10-pound weight in your hand and then bend your elbow, your body is going to use more muscle fibers to do that, and they'll also be shortening to their deepest extent. Once you have all that, again, the sarcomere shortening, the muscle fiber shortens. And what's required for the muscle to relax is ATP. So from the conversion of ATP to ADP, the cross bridges and the enzyme ATPase starts to break it down, and the heads return to their cocked position. So basically what happens is that a muscle will contract on its own, but to get a muscle to relax, you have to have ATP present, which is kind of the opposite of what you might think. You would think that, oh, well, it takes energy to contract a muscle. No, it takes energy to relax your muscles. And this next point kind of makes that. One of the reasons that after someone has died, they go into that phase of rigor mortis. There's a couple of reasons for it, and one of those is because you have this decreased ATP production. After you die, you're obviously not making more ATP, and so it starts to get really limited through the body, and without that ATP, your muscles just stay contracted. It takes energy to relax your muscles. So here is just kind of that cycle of it. You can kind of follow it along through the muscle relaxation where ATP binds to relax them, or the continued sustained contraction where you just have ADP present. Once ATP binds, like I said, the myosin will release, and then it can rebind to a deeper actin binding site, or myosin binding site on the actin. Here it is, you can see relaxed. There's minimal overlap on the fibers. There's no binding. There's that space between the myosin and the actin. And then in the contractile phase, you can see it's starting to shorten a little bit. Like if you look at this overlap here, it's definitely a little shorter. The myosin and the actin have bound together. And then in a full contracted state, that space right here is very, very much minimized. And you have, again, they're still connected at that point. So stimulus for contraction. There has to be a neurological stimulus for the muscle to contract. Acetylcholine, as I said, sometimes is abbreviated ACH. You'll see that AC capital, I think it's usually capital H. I might be wrong on that. But anyway, ACH, acetylcholine, that's a neurotransmitter that activates your skeletal muscle. It's produced in the motor neuron and then stored at the end of the axon in that synaptic vesicle. Acetylcholine gets released into the cleft after you get the impulse when it stimulates the motor end plate of the muscle fiber. Once the muscle receives that impulse, once the sarcolemma has been depolarized and it's received that impulse, the sarcoplasmic reticulum are going to release their stored calcium, and that's what's going to allow the troponin and tropomyosin to move out of the way. You have to have calcium binding to those regulatory proteins to get them to change their shape. Once they do that, then myosin can bind actin. Sarcoplasmic reticulum have a lot of calcium. They have a high concentration of calcium in there. They kind of pump it in. And so that's what allows, like I said, the exposing of the myosin binding sites. Once the binding sites are out of the way, the cross bridges can form. Myosin pulls on actin, and then that causes sarcomeres to shorten. The energy ATP, again, is allowing those myosin heads to release and rebind pretty quickly. As long as your body is sending an impulse to contract, it's going to continue to contract. So whether that is a conscious impulse of you holding something in your hand and not letting it go, or if there's, let's say you're having like a little muscle spasm, a muscle twitch, even that is, even if it's not a conscious thing, your muscle is still getting a neurological impulse. Once the nerve impulse stops, though, once that electrical impulse stops, you start to have a relaxation of your muscle. So now we're looking at the opposite event. How do you actually relax a muscle? It first starts, again, with the cessation of the nerve impulse. And then you have this enzyme, acetylcholinesterase, that gets released in the motor end plate and in that synaptic cleft to start to break down acetylcholine. So remember, enzymes are usually named. If you see ACE at the end of it, you can assume that it's an enzyme. And then usually whatever is in front of it, that's what it's going after. So acetylcholinesterase is going to break down your acetylcholine, and it's going to resorb it back into the motor neuron. Calcium is returned to the sarcoplasmic reticulum. ATP is used to pump all that calcium back into these storage tubes. And the ATP binds to the myosin heads so those linkages can be broken. Actin goes back to its original position, troponin and tropomyosin recover the binding sites, and the muscle can't contract. So here's the events listed out if you prefer to see a list rather than a picture of it. So you should be able to read through this, understand this, know the structures, know the stages, understand how they relate to each other. And this can be pretty intense. If this is the first time you've ever learned muscle contraction, it is a lot of things happening, and it probably took me 4 or 5 times of teaching it before I truly felt like, yes, I understand this very well. But that doesn't mean that it's always easy to understand if you are just now seeing this for the first time. So hopefully you have already done some pre-reading, you've watched some other videos on this stuff, and you are planning to maybe pause if you need to. Don't try to finish all this lecture at once if you can't. Go watch some other videos, take a break, and come back. And then the opposite, obviously, is the relaxation. Okay, so let's look at the energy sources for contraction. We've talked about ATP a little bit, but there are other ways our body can get energy. So energy from muscle fiber contraction usually comes from ATP, but it is in limited supply, so it has to be constantly regenerated. Our bodies only store enough ATP for a few seconds to maybe a minute or two. It really doesn't store it like it would other fuel sources. Creatine phosphate is another one. It's present to initially regenerate the ATP because it has those high energy phosphate bonds. It has this phosphate that your ADP is missing. And so we can use the breaking of this creatine phosphate to regenerate our ATP. Essentially, this only happens, though, when the ATP supply is high. Once you have a high supply of ATP, creatine phosphate, phosphokinase, creates a lot of, synthesizes a lot of creatine phosphate. Because if you have a high amount of ATP, you're probably not doing any rigorous activity. So it's a good time to go ahead and beef up your other energy stores. Whereas as you start to break down your ATP, that energy from creatine phosphate can then be transferred. So now you're using your creatine phosphate that you were making when your body was at rest. Now you're using that creatine phosphate to regenerate the ATP, and that's just because this can be a lot faster. If as it's saying right here, creatine phosphate gets really fastly used up, the cell must then go through cellular respiration to generate ATP. So really what's happening with creatine phosphate is that it's going to, again, we only have enough ATP for a couple of seconds of intense exercise, especially if you have to lift something really heavy, or, you're running from something. That couple of seconds of energy, rather than having to go through cellular respiration to make more ATP, your body at the muscle at the cellular level can just take this creatine phosphate and really quickly make ATP over and over and over again. But once that runs out, that only has a limited supply of creatine phosphate as well. So this is just a way to make sure your body has the energy it may need for emergency situations. Here it is, just kind of showing you that when you have a lot of ATP, your body is at rest. It's going to prioritize making creatine phosphate. Once your body is active, though, and it's using up its ATP, that's going to start breaking down that creatine phosphate to make ATP, to rephosphorylate ATP again, so that way muscle cycling can happen. Okay, if we remember, just a quick review of cellular respiration. So glycolysis is that first phase. It is anaerobic, so if you are running, and you're not getting enough oxygen, your body is still going to be able to go through glycolysis. It replaces a little bit of ATP, but not very much, because it is that incomplete breakdown of glucose. It only gives you 2 ATP per molecule of glucose. Once you start to get into the mitochondria, you can have aerobic respiration. It is a complete breakdown, the complete oxidation of glucose, but you do have to have oxygen for that to happen. It gives you 28 ATP per molecule of glucose, though. So you can see why once you start to get to the point of exercise where you aren't really able to take in oxygen as much anymore, your body kind of really does shut down quickly, because you're going from making 28 ATP per glucose molecule to just 2. To make sure that you have enough oxygen, we do have 2 oxygen-carrying molecules, hemoglobin and myoglobin. Hemoglobin you might be more familiar with. It's what gives red blood cells their color. They can store 4 oxygen molecules per hemoglobin molecule. So you can have 4 oxygens binding to every hemoglobin. Myoglobin is in your muscles. It is specifically storing oxygen in muscle tissue. So it increases oxygen availability. It's kind of like, hemoglobin has to be shared with the whole body. Your entire body is going to be able to use your supply of hemoglobin. Myoglobin, muscle, is only in muscles, so it's like their own private storage of it. The only issue with myoglobin is that it can only store 1 oxygen molecule as opposed to 4 that hemoglobin can. This is just an overview of cellular respiration, so if you need to review that, you can. But again, just remembering the product in is your glucose. Your ultimate product out is carbon dioxide. That's what we exhale, water, ATP. So now let's talk about oxygen debt. Let's say that you are working out, or you're running from something, or whatever it may be, and your body is having to use up a lot of energy. If that isn't the case, right? If you're just resting, then your body has enough oxygen for it. But if you have been really, putting in a lot of effort and a lot of exertion, you start to form what's called pyruvic acid, and then that turns into lactic acid. And a lot of people contribute lactic acid to the reason why their muscles are sore. It is not lactic acid. That it's not like your body stores that. Your body is very good about diffusing that out, taking it to the liver, and breaking down your lactate and your lactic acid. But people tend to think that's what's causing their muscles to be sore after like an intense workout. What's causing your muscle to be sore is that the muscle fibers are being damaged. You're actually tearing those muscle fibers. It's the cell death, and that is what is causing the pain. Your body gets that lactic acid right out of there, whether you stretch or foam roll or whatever. Your body's is not gonna let waste products accumulate. Once that gets back to your liver, though, that lactate gets back to the liver. ATP breaks it down and converts it back into glucose. But during strenuous exercise, all the oxygen that you're using to produce ATP, rather than converting lactate to glucose, starts to build up this oxygen debt that develops. So essentially, your body always likes to have, even, we talk about blood moving through your body, through your circulatory system, and then going back to your lungs, and then you exhale your CO2. Your body never, if you're at rest, never really uses more than 25% of the oxygen that it has available. Your body's always storing about or retaining 75% of the oxygen that it brings in. And that is just for emergencies. That is in case your body needs to start creating a lot of energy. If there's something going on, it has this reserve of oxygen. But if you've been exercising really strenuously, maybe you're starting to drop down. Instead of having 75% of oxygen, you only have 25% oxygen. And so it can take, again, several hours for your body to build back up that oxygen supply. So instead of being in the red where you have, this really, really low level of oxygen, you want to start kind of rising up and up and up, so you have your higher base level of oxygen. And it can take hours. So oxygen debt refers to the amount of oxygen that liver cells require to convert the accumulated lactate back into glucose. So it requires oxygen. It takes oxygen to reform your glucose. And so it can take several hours to be able to for that to happen. And this is that recovery period. How quickly can you recover? If you are a really highly trained athlete and you are good with your nutrition and your hydration and all of those kind of things, good physical training, then yeah, you can recover pretty quickly from an intense workout. If you are just a normal layperson and you try to do a really intense workout, it could take you all day. You might still feel bad the next day. And that's because you have such a high oxygen debt. Your body is putting so much energy into just trying to rebuild that oxygen debt that your whole self can suffer for it. So that's called your excess post-oxygen consumption, your EPOC. So it's basically kind of like saying, even after you've exercised, your body is still in that state of recovery for hours and hours and hours. So looking at muscle metabolism, this is a nice little chart for you to understand how does intensity compare to the type of energy that's going to be used, how much ATP is produced, and what waste product you have to deal with. So low, moderate intensity, you're basically using glycolysis. You're still making plenty of ATP, and really your only waste product is carbon dioxide because it's aerobic. High intensity starts to get anaerobic. You start to form a lot of lactic acid, very little glucose production, and then you have that lactic acid accumulating. Okay, so our muscles, as they're contracting and producing all this energy, they're also producing a lot of heat. So less than half of the energy released in cellular respiration reactions is used to form ATP. All the rest becomes our heat, which then we use to maintain our body temperature. When a muscle loses ability to contract, it starts to experience fatigue. And so that can be from electrolyte imbalances. Maybe your body is having trouble. It does not have enough electrolytes, salt, calcium, potassium, salt, chloride, calcium, potassium, those kind of things. And so you start to have this decreased ability to produce ATP. You can also have a decrease in pH from lactic acid that can start to increase your muscle fatigue. And then a muscle cramp. those can also start to happen. There's several reasons, several theories to why you might start to get muscle cramps. It could be changes in the extracellular fluid. So again, that change in the pH or the electrolyte imbalances, calcium availability. You could just have, again, an overstimulation from a motor neuron. That can absolutely happen. Just like a computer can glitch. Sometimes you have to turn it off and reset it. Same kind of thing can happen with your muscles. Sometimes they just have to, ice them down, get them nice and cold, some compression, and that is a good way to reset your cramps. So now let's look at the types of muscle fibers. So we're still looking at skeletal muscle, but every muscle, right? If you think about your hamstrings or your calf muscle or your biceps muscles, they are a blend of different types of muscle fibers. There are fast and slow fibers. So every muscle is going to have a blend of these two, but depending on the specific muscle you're talking about and depending on the person, that blend is going to be unique to each of us. So fast fibers, that's the majority of the muscle fibers that we have. Really quick movements, they reach fatigue quickly, they have fewer mitochondria, a lot of anaerobic metabolism. So essentially fast fibers are powerful contractions, short-term activities. They are not going to last very long. They fatigue quickly. Slow fibers are the opposite. They are not quite as strong of a contraction, but they are fatigue resistant. They use aerobic metabolism for endurance activities. We usually like to say people that are marathon runners tend to have a higher proportion of slow fibers, and people that are sprinters are going to have more fast fibers. If you do like a lot of Olympic powerlifting kind of stuff, you're really working your fast fibers. If you do more low weight, higher rep activities, longer HIIT workouts, things like that, you're probably working into your slow fibers. They also start to respond to changes in activity level. So as you train those muscles, they start to change and grow, or maybe as you don't use those muscles, they can also start to shift. So two conditions, hypertrophy and atrophy. So hypertrophy is when the muscle cell starts to get larger. It starts to actually increase in its size. Whereas atrophy is a decrease in the muscle size or strength. If you've ever been bedridden or broken a bone or something like that, where you had to be on bed rest or you had to rest for a long time, you can see, I had my arm in a cast for, a couple of months at one point, and it's like, oh yeah, my arm that was in the cast is much, much smaller than the other arm, and it's because, well, that arm in the cast wasn't getting used. Those muscles weren't being used, and when you don't use something in your body, it starts to break it. It starts to decrease, in this case, its size. So types of exercise determines how the muscles respond. Low intensity causes slow fibers to increase and they become more fatigue resistant. Forceful exercise starts to really work those fast fibers. It does not increase the number of muscle fibers, but it does increase the filaments, the actual proteins within those fibers. So whether you're doing hypertrophy or atrophy, whatever case you may be in, it's not that you are increasing the number of muscle cells or decreasing the number of muscle cells. This is all about size of the cells. If you were to increase the number of muscle cells, that would be hyperplasia, and that's like a tumor. There's only so much space in your arm, and so your biceps, if you start to increase the number of cells, that's increasing. That would be then like a tumor growing. But if you're just at the gym and lifting weights and your muscles are getting bigger, that's hypertrophy. All right, so looking at muscle responses. So this is kind of like in lab work, like trying to understand how muscles respond to different stimuli from the brain and the nervous system. So one method is to remove one muscle fiber, one single muscle cell, and then essentially attach it to some leads and send a certain signal strength to it at a certain time and see how the muscle fiber responds. So essentially, muscle fibers in a lab can be exposed to stimulus of varying strength. Maybe they just start, at 10% and then go up to 20, 30, 40, 50, as strong as the machine will start to go. But what they find is that muscle fibers are unresponsive until you reach what we call the threshold stimulus. And the threshold stimulus is the minimum strength needed to generate a muscle impulse. And then through that muscle impulse, the full muscle contraction. Because once a muscle reaches threshold, it's going to contract. But up until that point of a signal being strong enough to actually reach the threshold stimulus, it won't contract. The example that I always give is like, when I was a kid, Saturday morning, my mom would always like to sleep in. And as long as I was in the kitchen or if I was, out around the house, as long as I wasn't making too much noise, she really wouldn't wake up, right? I could make some noise and, as long as it wasn't too bad, she wouldn't get up and everything would be fine. But as soon as I made, enough noise, like opened a cabinet and started pulling out pans and she thinks I'm about to cook something or whatever it may be, she was going to jump up and, get to work, get to action. So it's that threshold stimulus for my mom. It was, the level of noise. What's the threshold? Okay, yep, I'm fine. I'm going to stay in bed. Oh, nope, now we're over that threshold. I got to get up. I got to do something. It's the same thing with your muscle fibers, except it's an electrical impulse. Really what's happening is that once the motor neuron impulse reaches that threshold, your acetylcholine will be released. And that is then, that sufficient amount of acetylcholine being released is what's going to stimulate the muscle fiber. So looking at muscle contractions and different types of muscle contractions, the response of a single muscle fiber to a single impulse, we call it a twitch. And a twitch is a full cycle of contraction and relaxation. And you experience twitches. If you're falling asleep and you're starting to like really just kind of doze off, and then all of a sudden you just give a little twitch, your kind of body just kind of jerks a little bit. That's a muscle twitch, right? They record that through a myogram. So that's recording the stimulus and the contraction. And what they notice is that after the stimulation, there is this brief period called a latent period, this little delay between the stimulus and the contraction. And that then is necessary because it allows the muscle to go through its cycle of relaxation, contraction, over and over again, so that way it can actually contract. When it does contract, it contracts to its fullest extent. With each twitch generating the same force. We call that the all or none response. So essentially what all this is saying is that once you have a muscle fiber stimulated to contract, it's going to contract fully. It's not like it only contracts a little bit. But this goes back to what I was saying, in that if you aren't holding something, if you don't have, let's say you have a twitch in your biceps. Well, if you're not holding something and it's just your arm, your biceps is only going to use a couple of fibers to do that. A small portion of its fibers to do that. But as you increase the weight of whatever you're holding in your hand, let's say you hold a pencil and then you hold a book and then you hold, a 20-pound weight, that increase in weight is going to cause more fibers to have to contract to their fullest extent. Until eventually you might be holding something that is too heavy, and even with all of your fibers generating at their fullest extent, you still can't hold it. But your body will always, once it contracts a muscle, will always contract it to its fullest, and that's the all or none response. Here's a little graph of that. So the force of contraction on the y-axis, the time that it's taking on the stimulus, on the x-axis rather. So you can see that latent period. There's the stimulus, and then there's this small period where it's traveling along the sarcolemma, down the T-tubules, into the sarcoplasmic reticulum. The calcium is being released. The troponin and trypamyosin are getting out of the way. Oh, now we finally have the myosin-actin heads binding, and we can contract. And so you have this kind of period of contraction from that one single stimulus, and then it starts to relax. And you notice that the relaxation period is much longer than the contraction period. That's because you have to get rid of all that calcium, reform the myosin, the troponin and trypamyosin binds with the actin. There's a lot more that has to happen. And this also allows our bodies to make sure that we don't go into sustained contractions. So what can happen through summation, though, is that you start to add all of those electrical impulses. So a muscle fiber receives a series of stimuli, increasing frequency until it's unable to relax completely. So we call that summation. So it's think about a bunch of small signals all being sent at a very short period of time. That causes the muscle to basically stay contracted. And what will happen is that the muscle fibers within a certain that a motor neuron activates will actually kind of cycle through. So even if you are, holding, let's say from the beginning of this lecture until now, you've been having your elbow bent with your hand at your shoulder. It's not like the same muscle fibers are working the whole time. Your body will cycle through different fibers. And as you add more weight, it'll add more fibers to that cycle as it needs to. Once you get up to a high enough frequency, the relaxation time becomes very short, and we call it partial tetany. And then if you have a really, really high stimulation rate, you can actually have a complete tetanic contraction. The thing to note is that uh, you can only reach complete tetany in a lab. It's required to have some type of external electrical signal to reach complete tetany. Our nervous systems are only capable of partial tetany. Here you can see some of that happening. The arrows are indicating the timing and the spacing of the different stimuli. And you see that as you shorten that time between the stimulus, it's not like you're maintaining the same force of contraction. They actually add up. So that's the summation. In that if you start with one stimulus, while the muscle is still partially contracted, if you do another and then another, you actually increase the total force of contraction. And then this one right here, this kind of solid straight line up at the top, that's supposed to be describing complete tetany, where the muscle is fully contracted at its highest, most, every fiber is staying contracted. That we can't really get as, in like I said, in our bodies. We can just kind of have this partial tetany where it's, maybe here where it just kind of is slowly maintaining this lower level of engagement, but it will eventually have to relax. So I've been talking about this recruitment without trying to really say this word very much, but we can talk about the recruitment of motor units. So a motor neuron and the muscle fibers it controls make up a motor unit. So a motor unit is one motor neuron and then multiple muscle fibers. It really depends on where in the body you're talking about, how many muscle fibers will be in a motor unit. If we're talking about your eyes, that's such delicate, fine movement, the motor units are really small. Your motor neuron might only be stimulating 10 muscle fibers. But if we talk about like your hamstrings or your leg muscles, you might have one motor neuron stimulating 1,000 muscle fibers because it's not very fine control is needed. So just kind of think of that, a motor unit is a motor neuron and its muscle fibers. Increasing in the number of activated motor units within a muscle at higher intensities is called motor unit recruitment. So that's what I was saying about, let's say you're just holding something, you got something in your hand, you're just holding a pencil and you flex your elbow, you bend your elbow so your hand's at your shoulder. Okay, it might recruit 1 or 2 motor units. But now replace that pencil with a 20-pound dumbbell and it's going to have to recruit a lot more motor units to be able to overcome the weight of the dumbbell and still contract like you want it to. So recruitment causes an increase in the strength of a contraction, and a muscle achieves maximum tension when all of its motor units have been recruited. So unless you're at the gym and you're doing your PR, personal record, max weight lifts, you're not really going to be reaching maximum tension. For most of us, unless you just tend to go all out in your daily life and you're trying to bench press carbs or something, most of us are just using some of our motor units, not all of our motor units. Here's a depiction of that. So the blue is motor unit 1 and the red is motor unit 2. You can see each of these fibers is kind of coded. So they're mixed around each other. You have some of the blues, the motor unit 1, or the blue ones, they're kind of interspersed in here with the red ones. And when you get a normal, a standard contraction to pick something up, maybe at first it's just motor unit 1, and then your body's like, oh no, this is heavy, we need help. And so motor unit 2 kicks in and you start using all of these fibers. So summation and recruitment produce a sustained contraction of increasing strength. Again, your body will continue to send signals of increasing frequency as well as gathering the motor units around it to help hold that contraction and make it stronger. This actually does, it happens more frequently than you think because this is how we maintain most muscle tone and our posture. So muscle tone is a continuous state of contraction with a few motor units at a time within a muscle, even when you're at rest. So again, let's say there are 5 different motor units. While you're just kind of sitting here listening to this lecture, your body is going to be cycling through those different motor units just to help hold you in place, just to help maintain maintenance and posture. If you were then to add weights or start moving around, your body recruits more motor units because at that point you're no longer just holding posture, you're actually actively moving. There's different types of contractions. So looking at the ways that muscles kind of can generate force. They don't have to shorten. Your muscles can go into isotonic or isometric contractions. An isotonic, this is when you're actually shortening the muscle. This is what you would normally think of as a contraction. You know, you're bending your elbow, you're flexing your hips, you're straightening your knee, standing up. That is a contraction with movement. The muscles are actually changing length as they generate force. Isometric, this is generating force without shortening. So holding a weight in one position, resisting overstretching, opposing gravity, you know, just standing in place. That is a isometric contraction. Many movements combine both types. Many movements are gonna be both isotonic and isometric. Okay, so now let's look at smooth muscles. That was kind of most of the skeletal muscle stuff. Let's start to look at smooth muscle a little bit. So smooth muscles, elongated and tapered ends rather than the kind of fiber, straight and narrow shapes of skeletal muscle. They have these tapered ends. And underdeveloped sarcoplasmic reticulum, so not going to be great at sending out calcium as much. They do still have thick and thin filaments, but they don't have the striated arrangements that skeletal muscle does, so they have this random arrangement to them, and so they contract in a random pattern. There are multi-unit smooth muscle and visceral smooth muscle. Multi-unit in your blood vessels, the iris of the eyes, they occur separately instead of as sheets. And visceral smooth muscle occurs in these sheets of tissue, in the walls of organs, things like that. And so that's what allows them to accomplish peristalsis. It's because it's this big sheet of muscle rather than these individual little muscle fibers. But it makes sense in the iris because, you know, in blood vessels and in the iris, the contraction dilation that is required for those structures needs to be very specific and very localized. Whereas, you know, with visceral smooth muscle, okay, you're just kind of pushing stuff along. It does not have to be quite as specialized. So it does have let's look at the similarities and differences, the comparing and contrasting smooth muscle. So similar to muscle contraction, skeletal muscle, in that both involve the reaction between actin and myosin. So they're both using actin and myosin for their contractions. And they're stimulated by membrane impulses. They require calcium ions and they use ATP. So those things are still happening. You still have this electrical stimulus that's coming. You're still having a release of calcium ions for the contraction to actually occur, and you need ATP to relax. Some of the differences is that smooth muscle can be stimulated by acetylcholine and norepinephrine, which is another neurotransmitter and hormone. It works as both. Whereas skeletal muscle is only stimulated by acetylcholine. Hormones can stimulate or inhibit smooth muscle action, but don't really affect skeletal muscle. Smooth muscle is a lot slower to contract and relax. It takes sometimes several minutes for these things to happen. And it contains, smooth muscle maintains longer contractions with the same amount of ATP. It also, smooth muscle can change length without the change in tautness. So it can kind of shorten and lengthen without contracting. Smooth muscle is different, y'all. Okay, cardiac muscle. Cardiac muscle only found in the heart. It does have striations, but instead of being straight lines, it branches and kind of forks at certain places to kind of create this interconnected network. Mechanism of cardiac muscle is the same pretty much as skeletal and smooth. The differences are that, again, you don't have these developed sarcoplasmic reticulum, so it doesn't really store much calcium. The transverse tubules supply that extra calcium from the extracellular fluid, and so that's where they're getting theirs from. That's why calcium levels are very important in the body, and your body will break down your bones as it's needed to try and supply calcium to your heart, to your muscles. Cardiac muscle is also self-exciting and rhythmic. It's autorhythmic, and it creates this consistent pattern of contract, relax, contract, relax, contract, relax. It has a pacemaker function. You might have heard of people getting external pacemakers. It's because their heart can't maintain that self-exciting rhythm, and so they have an external pacemaker that sends these signals to make sure that the heart kind of keeps up its natural rhythm as it's supposed to. And they also have these things called intercalated discs. And intercalated discs join cells together, so that way they're contracting at the same time with the same force, and it actually increases the overall force of contraction between the cells. And when we get into the cardiovascular chapter, we'll look a lot more at the heart, its muscles, and how it functions. So this next table, it just compares skeletal smooth and cardiac muscle with its locations, functions, characteristics, how it's controlled, all those kind of things. You should be familiar with this chart. Hopefully this helps you to kind of organize some of it. Okay, now let's just look at a couple of slides on skeletal muscle actions. So with muscle attachments, skeletal muscle attachments, you may have heard of origin and insertion. These are both attachments. Attachment is just a general term. Origin is the less movable portion, whereas the insertion is the more mobile end. Typically, the origin is closer to the trunk. It's more proximal, whereas the insertion is going to be more distal. And when muscles contract, they're usually going to pull the insertion toward the origin. That doesn't always have to happen. You can have reverse functionality where you actually move the origin towards the insertion. So it can go back and forth. And I don't always love looking at muscles in terms of origin versus insertion and learning them that way, but it is a way to organize it, really understanding that there are multiple attachments for a muscle. And some have more than just one origin and insertion. Example of that is the biceps brachii. Biceps, bi is two, sep refers to cephalic, that's your head. So the biceps has two heads, and then brachii is your brachium. So that refers to your upper arm. So biceps brachii is its full name. If you just say biceps, you actually have a biceps muscle in your hamstring, your biceps femoris. And so when you're talking about the muscle in your arm, you really should say biceps brachii to be specific about that. Its two attachments are on the scapula, the coracoid process, and the supraglenoid tubercle. So it's just a fun name to say. So it has those two heads that start up at the shoulder blade, and then the insertion is on the radius in the forearm. The belly of the muscle is kind of running across the humerus, the bone in the upper arm, and it's one of its main actions is to flex the forearm at the elbow, but it also does shoulder flexion and some rotation of supination and pronation in the forearm because it crosses all of those joints. So whatever joint a muscle is crossing, it then will have actions at that space. Here you can see the biceps muscle right here on the front. Deep to it is the brachialis, and then on the posterior surface of the arm is the triceps brachii. So I will let you decide what triceps means. You can see the origins. This is the short and then the long head of the biceps. And then it forms, those two heads come together into the belly of the muscle, and then you have this one tendon attaching or inserting. Same thing. So for body movements, we've said that the function of skeletal muscle is movement, maintenance of posture. And so the bones act as these levers for movement. The bones, if we didn't have bones, you would just be like a jellyfish, kind of flopping all over the place. And so by having both muscle and bone, we can move and move in a direction, push forward and create force. Bending and straightening the upper limb is an example of that flexion. And when we look at that, we can think about the different parts of a lever. A lever is a mechanical system, and so we can look at our body in that way. You have the rigid bar, the rod of the forearm bones. You have the fulcrum, which is the elbow joint. That's the point of leverage. And then you have the object being moved against resistance, which is your hand. Maybe your hand has something in it, maybe it's not, but it's still that most distal portion, so that's going to be where the weight is. The objects supplying the force would then be your anterior arm muscles, mostly your biceps brachii, but also a couple others, just so you know. Rarely one action is just one muscle is responsible for an action completely. Usually there's multiple muscles that are going to be working. That's that recruitment idea again. As the biceps brachii contracts, it pulls on its inserted tendon in the radius, and that then flexes the elbow and causes the hand to be moved against resistance. Resistance being gravity, right? Just the weight of the hand and gravity. So here you can see that action happening. You have your biceps brachii contracting muscle, generating force. The elbow acts as the fulcrum, and then the hand is the resistance. And then as you start to extend the elbow and you start to let that back out, now your triceps is contracting. The fulcrum is still the elbow, and then you still have the resistance at the end. So changes in the angle and opposing movements between bones at a joint, things like flexion and extension. Flexion is decreasing the angle because you're closing it off, whereas extension is increasing the angle, like when you straighten your arm. Those are just two general times. Actions are almost always paired, so just be noticing that when you learn one, you want to be sure you're learning the other. And usually muscles function in groups. So like this example right here, what we're seeing is we're kind of focusing on the biceps muscle contracting. But even as the biceps muscle is contracting, your triceps has to stay relaxed, but it still has to allow for some engagement to help stabilize. And same thing here. Yes, your biceps is contracting, but if your triceps is contracting to pull this lever down, oh, that wasn't a good one. Let's stay up here on this one because I didn't realize I had that pulley. So stay up here on this one real quick. So this movement that we're seeing is this flexion movement of the forearm. But let's start to say, okay, now we're starting to lower this down, right? Instead of flexing the elbow in this one, we're lowering and extending the elbow. Well, even though your biceps is lengthening and in your mind you might think, oh, it's not really working, it still takes effort for the biceps to hold this weight, to hold this resistance, and not just let your arm fall completely, right? When you go to extend your elbow, rarely do you just let it go. Typically, you straighten it slowly and with some type of control. And so the biceps is still having to contract and engage to make sure that you don't just drop your hand. And so usually when muscles are working, they're working in groups. The muscle that causes an action, the one that's doing most of the work, is your agonist, your prime mover. Muscles that are assisting it are called synergists, and the ones that oppose it are called an antagonist. So you have an agonist doing the action, synergists are helping out, and antagonists are opposing. In the example of elbow flexion, the agonist, the prime mover, is your biceps. The synergist would be some of these deeper forearm, upper arm muscles like your brachialis. And then the antagonist is the triceps. And in this example, it's nice because it's telling you, oh, well, this is your relaxed muscle. So if the bicep is going to contract and you're going to flex your elbow, the triceps has to relax to allow that action to happen. So that's how you can usually tell which one's an agonist and which one's the antagonist. When one muscle engages, what's the other muscle that has to relax to allow that to happen? Because if the antagonist, in this case, the triceps, if the antagonist didn't relax, you wouldn't be able to flex your elbow. Your elbow would stay locked extended and it would just be your biceps and triceps fighting against each other. And same thing here. If your biceps didn't relax while you were extending the elbow and it stayed contracted, then your biceps and triceps would just kind of fight each other. So the antagonist has to relax while the agonist is engaging. And I think that's it for this lecture. All right, have a good one.